This is podcast number 38. We're going to wrap up here the oxidation of glucose and how you're going to make ATP. So far we haven't made a whole lot of ATP. In this one we're going to make lots of ATP. So if we were face to face, I would just flash this up and then I would say memorize everything on this slide and go on to the next slide. But I'm just kidding. This is just to give you an idea that metabolism is, in general, pretty complex, but we understand a lot about it, and it's also very interconnected. So if we look at a big diagram like this that looks at what is called intermediary metabolism, this is basically how cells turn food, molecules, compounds into energy, and synthesize different compounds like amino acids, for example. If we zoom in, you can see part of the citric acid cycle here and the 2-carbon acetyl-CoAs going into this cycle, so hopefully you recognize that sort of stuff now. Now we're going to, it's very important to know where things are happening, and so I want you to be able to draw a simple diagram of a mitochondrion. A lot of people complain about their ability to draw, but I know all of you can draw circles, ellipses, lines, because you can write. And if you can do that, you can draw a mitochondrion. So this is what I want you to do. If I, draw something super simple like this. Okay, now I'm drawing with my finger, a little bit tough for me to do this, but one membrane on the outside and on the inside, a folded up membrane like that, okay, and the outside of this space would be considered the cytoplasm. This innermost space inside both of the membranes is called the matrix. M-A-T-R-I-X. Okay, and then this is the inter with a T membrane space. I'm not going to write out membrane space because it takes me forever to write with my finger and the trackpad, but this is the intermembrane space, not inner, inter with a T. So intermembrane space between two membranes, okay? These spaces are really important in when we're talking about metabolism, where things happen, okay? So stage 1 happens out here in the cytoplasm. That is the glycolysis. Stage 2, Roman numeral 2 here. This starts in the cytoplasm and then it moves across both membranes, okay, to end up in the matrix. So it starts in the cytoplasm, moves across both membranes to the matrix. Now when you're drawing this on a test, it's important that you give me some words here. Starts in the cytoplasm, moves to the matrix, because otherwise it looks like you're saying the entirety of stage 2 happens in the matrix, and that's not true. Starts in the cytoplasm, moves to the matrix. Stage 3, Roman numeral 3 here, that happens in the matrix. That's the citric acid cycle. Stage 4 happens on the inner membrane, okay? So here's the outer membrane, here's the inner membrane. Stage 4 happens on the inner membrane. And then stage 5 is going to move from the intermembrane space into the matrix. Hydrogen ions are going to move from the intermembrane space, that's supposed to be an H, plus, are going to move from the intermembrane space into the matrix, okay, during stage 5. That's supposed to be Roman numeral 5 here. Okay, so on a test, if I ask you to say where did the stages happen, you're going to draw a very simple diagram of mitochondrion, just one line for a membrane, not two lines for a membrane. One line equals one membrane. One line for this membrane, one line for that membrane, two membranes in a mitochondrion. The space inside both of those membranes is the matrix. The space inside the outer membrane, but before the matrix, is called the intermembrane space. And then outside the mitochondrion is the cytoplasm. Okay, so lots of you are going to ignore this vice. You're going to look up a picture of a mitochondrion. You're going to try to duplicate that, and you're going to try to draw something fancy like this 3D mitochondrion where you take out a slice and that sort of thing. Do not draw that. I have looked at thousands of drawings of students, and I can tell you that most of you are not good enough with your drawing skills to draw that accurately, and it's confusing for me to look at as to what you're trying to say. I want you to know where things are happening in the mitochondrion, so draw a simple mitochondrion. Okay, let's go forward here. All right, so we've gone through stage 1, stage 2, stage 3, citric acid cycle. Now we are going to get some of the energy from those molecules that we stored energy in up to this point. So we used NAD as an electron carrier to store high energy electrons in the form of NADH. And we use FADH2 as another electron carrier to store high energy electrons. And now we're going to use those molecules in stage 4 to get some energy back. So if you measure the pH of a mitochondrion, when you add the NADH, the pH goes down inside the compartment, the innermost compartment of the mitochondrion. Okay, this indicates, so pH is the concentration, the negative log of the hydrogen ion concentrations. So as pH goes down, hydrogen ion concentration goes up. So as NADH gets oxidized, taking the hydrogen off, it's going to be moved into one of the compartments of the mitochondrion, and we'll talk about that. Okay? If you prevent hydrogens from moving across the mitochondrial membrane, you don't get much of a pH change. But if they can move across compartments, those compartments we just talked about, you get a big pH change, relatively big pH change. Okay. On the inner mitochondrial membrane, okay, it says outside mitochondrion, inside mitochondrion. This is just the matrix is on this side. This is the inner mitochondrial membrane, and then out here is the outer mitochondrial membrane. So I don't like that your book says outside the mitochondria. It's not outside the mitochondria. It's just outside the inner membrane. So this is the intermembrane space, and this is the inner mitochondrial membrane here in pink. Okay, there are proteins embedded in that membrane, a lot of them. 90% of that membrane by weight is protein, and they carry electrons. And they move electrons from one electron carrier to the next in a chain, like a bucket brigade. And as the electrons move from one to the next, they lose energy. Okay, the electrons from NADH come off NADH, leaving NAD, and they get passed on to complex 1. Complex is just a bunch of proteins. And then those electrons move to complex 2. So when NADH becomes NAD, the electrons get taken off NADH and put onto complex 1. NAD is oxidized. Okay? And that's a negative delta G, because it is now making that energy available to do work, and it's a spontaneous reaction. All spontaneous reactions are negative delta G. At the same time that electrons are passed from the NAD to complex 1, the work that is done is to move protons against their concentration gradient. So they move from the matrix to the intermembrane space, and that reduces the pH in the intermembrane space, just like we saw in that graph, because more hydrogens lowers the pH. Higher pH means less hydrogens. So hydrogens move across the membrane into the intermembrane space. FADH2, those electrons, same sort of thing. They're going to come off FADH2. FADH2 gets oxidized into FAD. Those electrons get passed on to complex 2. They're just entering the chain at a different place. And that energy that is released, the delta G that is can be used to do work, does the work of moving even more protons into the intermembrane space. Okay, from the matrix into the intermembrane space. Okay, and then the final place that the electrons go, so the electrons are losing energy as they go down this chain. This is like going down a waterfall. They're losing energy as they go down this chain. The last place they go is to oxygen. Now this isn't oxygen you breathe. This is molecular oxygen. So the oxygen that you find in the atmosphere is in the form of 2 oxygen molecules, or O2. Molecular oxygen is in the form of 1 oxygen molecule. It's unusual to find it in that form, and so it's written as half of an O2. Okay? They get passed on to that O2. O2 gets reduced by those electrons and becomes water. So the reason that you need oxygen, the reason your cells need oxygen, the reason you breathe oxygen is for this, is for some place for those electrons to go. If there's no oxygen available, if you run out of oxygen, stage 4 backs up and you can't do oxidation of FADH2, you can't do oxidation of NADH. Not only that, but stage 3 backs up and stage 2 backs up. And the only thing you have left that works is stage 1, glycolysis. So as I mentioned in the last podcast, normally if I were giving you this face-to-face, the glycolysis sheet I would give you is blue like you're running on oxygen, and that sheet still works without oxygen. And then stage 2, 3, 4, 5 is on a pink sheet, and they all require oxygen, and it's because of this, that the last thing to accept the electrons is oxygen, okay, and making water. So this whole stage here is stage 4, where you take the electrons off of NADH and FADH2, you pass them down an electron carrier gradient. They lose energy along the way, and that energy is used to move protons against their concentration gradient from the matrix into the intermembrane space. So the outside of the mitochondrion is way up here, and this is the intermembrane space. Okay? And then the oxygen that the electrons get passed on to is called the terminal electron acceptor. The last thing to accept electrons in the oxidation of glucose is oxygen. That's the last place electrons go. We've been passing along electrons the whole way that we're talking about this. The last thing you pass them off to is oxygen, which makes water. Oxygen is the terminal electron acceptor in mitochondria. Okay, and this oxygen is consumed, and excuse me, that makes some leftover hydrogens, and I'm sorry, it uses up some hydrogens, and that will make the pH even higher in the matrix. So then we have a pH gradient, more hydrogens in the intermembrane space, that means a lower pH, less hydrogens in the matrix, that means a higher pH. Okay? Because you now have a pH gradient, you have potential energy. There's more hydrogens on this side of the membrane than on this side of the membrane, and you're going to use those to do work in stage 5. We saw this sort of thing when we were talking about how non-cell vesicles, so just a membrane, vesicles, when we're talking about early cells, how they could make a pH gradient by moving hydrogens from one side of a membrane to another that reduces the pH inside the membrane. This is very similar to what happens in stage 4. Okay. Now, you have probably heard in the news that if people use, say, something that burns a fuel, like a heater or something like that, and they're not well ventilated, it can produce carbon monoxide, carbon with 1 oxygen, versus carbon dioxide, which is what you breathe out. Carbon with 1 oxygen, carbon monoxide, poisons electron transport. And where does it poison electron transport? Well, if you add carbon monoxide to complex 1, electron carrier 1, not much happens. It's still close to 100% of its activity. Same with complex 2, same with complex 3. But in complex 4, you see activity goes way down as you add carbon monoxide. So complex 4 is where electron transport stops if carbon monoxide is there. And this is how carbon monoxide kills you, is it binds to complex 4. You can't get much ATP out of your glucose, and you need ATP to live. You're only stuck with glycolysis, and that's not enough for your whole body to live, and so you die. And you can't smell it, you can't detect it. This is why you should have a carbon monoxide detector in your house that beeps if the carbon monoxide levels increase. So you just go to sleep. Okay, stage 5. This is where you're actually going to really make some ATP. There's this protein complex called ATP synthase that is sitting in the inner mitochondrial membrane. And what it does is, as those hydrogens from the intermembrane space that build up, they want to move back downhill because there's lots more hydrogens now in the intermembrane space than there is in the matrix. They want to move back downhill because there's a pH gradient. They move through this complex, and as they move through this protein complex, it's like they are going through a turnstile, like you would be going to a Browns game or an Indians game or a concert at a big venue, and you turn that turnstile. It turns this machine around, and as the machine gets turned around, it grabs ADP plus P and literally smashes them together to make ATP, and out pops ATP. So the hydrogen ions move down their gradient, okay, through this complex from high to low, and that causes this part of the molecule to spin around in a circle. As it spins around in a circle, it changes shape, and that shape change is used to smash ADP and P together to make ATP. There's all kinds of cool movies of this online. There's some links in the next slide. Just to save time, I'm just going to show you a couple diagrams. There was a key experiment done by a graduate student to prove that this thing was actually rotating. That was a hypothesis, but was it actually rotating? So what they did was they took this molecule, stuck it to a slide, and then they added essentially a propeller, a big piece of fluorescent actin on this part, and they tried to see, can we see that spin underneath a microscope? And these are stills from that movie. You can see it spin, and it spins 120 degrees at a time. So it moves from here to here to here, here to here to here, here to here to here. It moves 120 degrees at a time. And every 120 degrees it moves as it goes around in a circle, it spits out ATP. So that's where most of your ATP comes from, is that last stage, stage 5. So in stage 4, you pushed a bunch of hydrogens underneath the outer membrane of the mitochondria and into that intermembrane space. Then you use all those hydrogens to move downhill. This is sort of like water going through a windmill. And then as they push this rotator around, those hydrogens moving around, that is used to push ADP and P together to make ATP. And most of the ATP you get out of a glucose molecule or any other kind of molecule comes from this process, and it requires oxygen. So how good is it? If you looked at the energy that is potentially in a glucose molecule, how much energy are you actually going to get out of it compared to what you could get out of it? And that is an indicator of efficiency. If you measure how much energy is in a unit of gasoline, and when you burn that gasoline in your car, how much of that are you capturing in terms of forward motion, the ability to do work in your car? Car is about 20% efficient, in that about 20% of the energy that's in that gasoline actually moves your car forward. The rest gets transferred into heat. That's why your car has a radiator, so it can dissipate this excess heat. So what about these other molecules that we've made in stage 1 through 4, or before stage 1 through 4? How much energy do you get out of them? Well, FADH2, all the energy in there, when you oxidize FADH2 and you pass off those electrons to complex 2 in the electron transport chain, you get about 24% of the energy that is captured in the ATP you make. NADH, you get about 36% of the energy captured. Glycine, if you're going to, we haven't talked about this yet, but we will in the next podcast, how you break down stuff that is not carbohydrate. So glycine is an amino acid. If you're breaking down protein, you get about 38% of the energy. Lactose is what we started with in this series when we're talking about carbohydrates. Lactose is broken down first into galactose and glucose, and then we follow glucose throughout the whole system. And you get about 36% of the energy. Palmitic acid, about 35% of the energy. So where does the other energy go? It goes into heat. So you can, although this is a little bit controversial still, the heat that you make, you can harvest some of that heat, and it contributes to why you are warm-blooded, why you have an internal body temperature that is high compared to your environment. That's what you do with this waste heat. And when you are breaking down lots of molecules, okay, like when you're exercising, you generate more heat, and you have extra mechanisms to get rid of that excess heat, like sweating, for example, or breathing heavy, for example. Okay, now we're going to go over the handout sheets, starting with stage 1 and going all the way through stage 5, so you can see it all at once, sort of as a review. Okay, so stage 1, this would be blue. Blue because this happens in the cytoplasm and it can happen without oxygen. And the stuff that is underlined are the names I want you to know. Okay? So glucose going to glucose 6-phosphate. That takes an ATP, and that phosphate gets added on to the number 6 carbon. Then glucose 6-phosphate gets rearranged into fructose 6-phosphate. It's another 6-carbon molecule, just a different shape. That fructose 6-phosphate then becomes fructose 1,6-bisphosphate. Now you have a phosphate on carbon number 1 and carbon number 6. Since there's 2, we call it bis. And the extra phosphate came from another ATP. So I've rearranged this molecule. I spent 2 ATPs, and I have oxidized it as I went from glucose to fructose 1,6-bisphosphate, but I haven't gotten any usable energy out of it yet. Now I'm going to split that molecule into 2 glyceraldehyde 3-phosphate molecules. Okay, that glyceraldehyde 3-phosphate molecule is going to get converted to 1,3-bisphosphoglycerate by just grabbing a phosphate out of the cytoplasm. Remember, we're going to go through about 500 pounds of ATP a day. That means a lot of phosphate is floating around in the cytoplasm, and I can grab one and add it on to this molecule. So on carbon number 1, on carbon number 3, bis, 2 of them, phosphoglycerate. As I went through that enzymatic reaction, I also charged up an NAD, became NADH, and now you know that those NADHs are going to get used in stage 4 to help create that proton gradient, that H plus gradient, where there's more H plus in the intermembrane space and less H plus in the matrix. Then I'm going to take that 1,3-bisphosphoglycerate and take one of those phosphates off and add it on to ADP and make ATP. Since this happens twice per glucose, I made 2 ATPs. I spent 2 ATPs up here. So at this point, I am net zero for ATP, but I also have 2 NADHs, which later on is going to make me some ATP. Then I move that phosphate from the 3 position to the 2 position, and then I rearrange that 3-carbon molecule, and then finally I make pyruvate, and in that last step I also make an ATP. So if glycolysis is all I've got, because there's no oxygen around, I make 4 ATPs total. I spend 2 ATPs, okay? And those 2 ATPs I spend come away from the 4 I made, so I have a 2 net gain. I also made 2 NADHs. If there's no oxygen around, I'm not going to have much use for those, except you do use them if there's no oxygen around when you make lactate, but that's another class. Okay, so this all happens in the cytoplasm. This all happens stage 1. This all happens without oxygen. Now in stage 2, we're going to move into the mitochondrion, and this sheet would be pink if it wasn't black and white, because everything on this sheet requires oxygen. Not directly in the reaction, but if there's no oxygen around, it backs up to stage 1, and you can't do anything on this sheet. The cell doesn't do anything on this sheet. So everything on this sheet doesn't happen unless there's oxygen around. That pyruvate molecule has got to get from the cytoplasm into the matrix. And so it starts in the cytoplasm and moves across both membranes of the mitochondrion, the outer and the inner, and ends up in the matrix. Carbon dioxide comes off of that. That 3-carbon molecule becomes a 2-carbon molecule. It gets attached to coenzyme A. Coenzyme A are molecules that help enzymes do their job. A lot of vitamins are coenzymes. In that process, you also charge up another NAD. NAD becomes reduced, and then you're going to see that we're going to use those NADs in stage 4. So in stage 2 happens twice per glucose. 2 NADH is made per glucose, 2 CO2s removed per glucose, and those CO2s are breathed out in your breath. Then we're in the matrix of the mitochondrion, and in the matrix of the mitochondrion is stage 3. It's called the Krebs cycle, or the citric acid cycle, or the TCA cycle. They all mean the same thing. They're all stage 3. And that 2-carbon molecule, the CoA, comes off. It gets added to a 4-carbon molecule. 4 plus 2 equals 6, which is a 6-carbon molecule, citric acid. That citric acid is going to get rearranged into a bunch of different kinds of acids. And on many of these steps, 1, 2, 3 of these steps, you're going to make a reduced NAD molecule, make NADH. In one of those steps, you're going to make a reduced FAD molecule, FADH2. And in one of those steps, you will make ATP if you're a plant and GTP if you're an animal. And then you're also going to get rid of a couple of carbon dioxides as the molecule goes from 6 carbon to 5 carbon to 4 carbon. At the end of this cycle, that 4 carbon molecule is added to another 2 carbon molecule. You go around stage 3 2 times per glucose. So per glucose, stage 3 gets you 1, 2, 3, 6 NADHs, 2 FADH2s, 2 ATPs or 2 GTPs. Most of what you get out of stage 3 is those highly reduced electron carriers. Not a whole lot of ATP yet. In fact, we haven't made a whole lot of ATP at all. Now, all those NADHs and FADHs, H2s, we're going to use them in the electron transport chain. This happens on the inner mitochondrial membrane, where this is the matrix side, this is the intermembrane space, and way over here would be the outer mitochondrial membrane. So this is the space between the two membranes, the intermembrane space, not inner, not two N's, not INN, but INT, intermembrane, between membrane space. Okay. NADH passes off their electrons to the first electron carrier. FADH2 passes them off to the second electron carrier. And then as those electrons move down the chain of electron carriers, they lose energy, and that energy is used to move protons, hydrogen ions, from the matrix to the intermembrane space. So the proton concentration in the matrix is going down. The proton concentration in the intermembrane space is going up, which means pH is going down, because pH is the negative log of the hydrogen ion concentration. As the hydrogen ion concentration over here goes up, the pH goes down. And now you end up with a gradient of lots of hydrogen ions over here, not as many over here across this membrane. And then you use that gradient in stage 5, where they go through the ATPase. The hydrogens fall downhill back to the matrix. As they fall downhill, they go through this complex, which spins around in a circle, takes ADP and P, and smashes them together. Okay? How many ATPs do you get out of this last stage? Well, theoretically, you can get 36. I don't necessarily ask people to remember that number because it all depends on what else the cell is doing. So just like a car has a mileage sticker when it's brand new at the dealer, there's always a little asterisk on that and says your mileage may vary. So it depends if your car is going up a hill, depends on your car sitting in traffic a lot. Those numbers that are on the car are best case conditions, and the number of 36 ATPs made per glucose is best case scenario. If the cell is doing other things, like it's synthesizing stuff, or the cell is a little bit leaky, has a little bit of problem with its membrane, it won't get a total of 36 ATPs. But lots more ATPs out of this last stage, stage 5, than in any of the stages before this. And so if you can't do anything on this sheet, which is normally pink, and all you're stuck with is this sheet, which is normally blue, because there's no oxygen around, you can't make much ATP, and you can't do much, because there's not much ATP there. Okay? So if you poison electron transport in the last complex with carbon monoxide, and you shut down everything on this sheet, you don't have enough ATP to survive, and that's why carbon monoxide is a killer. Okay, so that's a review of glucose from the beginning to the end. In the next podcast, we'll talk about what happens with other types of macromolecules like proteins or fatty acids.